So tomorrow is exam 4, right? In lab time. What I'm going to do is that's going to be available from, let me write this out. Exam 4 will be available from 9:30 to 12 o'clock. So that's going to give you 2.5 hours. So that'll be an extra hour of time to work on it. So I don't know if he's going to have you do anything for the first 20 minutes. If not, sleep in, whatever. Do what you want to do. And it'll become available on my Canvas, the lecture Canvas at 9:30 for you to take a look at and start going on. And then it'll be due at 12 o'clock. So you'll have 2.5 hours. Hooray. So that means you have extra time to cry over the synthesis problem that maybe you didn't have in the last couple. So, as always, there is mechanisms, synthesis, predicted products, and then nomenclature. The nomenclature part is very minor because we don't really have any new functional groups to do this time, so it's minor stuff. And it's not really its own section, it's integrated into some of the other things. And the nomenclature, by that I mean like isopropyl bromide. Like, if you can't figure that out, then we've got some trouble. But that's the real nomenclature, so I'll just put that in parentheses because it's not really the big part here. So again, most of it is enolates. And the rest will be carbohydrates. And I think I originally quoted you guys at about 75% enolates, 25 carbs. It's more like 85-15. So if that helps for whatever reason, let that be so. The stuff we do today, of course, will not be on the exam because it would just be terribly unfair to do that. So that stuff, the amino acids, will just be on the final. Speaking of the final, I did put up a practice ACS final. It's only 105 material because, like I said, I made this practice for my 105 class, and so I'm just using the same kind of practice one twice because it is a big pain to make them. So, because of all the images and structures that need to be put in. So, that's on Canvas. You can take it as many times as you want so you can get used to the timing and make sure all the images are loading and everything. So, definitely check that out. So, that's on Canvas for you. And let's see, anything else to go over? Your synthesis problem, by the way, tomorrow does have a requirement that you use a certain molecule as all of your carbon sources. So, like, remember that first time it was propyl bromide and the second time it was butyl bromide? It is not an alkyl halide this time. But suffice it to say that make sure you follow that instruction. It's in bold. I've said it like we've done it on exam 3. I've talked about it after exam 3. I'm talking about it now. It's written in bold on your exam tomorrow. So make sure for your synthesis problem that you follow the directions that are given. So a couple of y'all still on the like fake exam 3 synthesis, like the extra one we did, still didn't follow that. So make sure that if there are questions, you ask them. So I will be around. I'm going to be having a review session for 105 at some point in that time period, but I'll be able to pop in if you ping me. If you at me, I'll see it and I'll be able to respond pretty quickly. So if there's any questions at all, let me know, okay? So other than that, it's like Just follow the directions, okay? Um, let's see. Anything else about the exam? No, not really. Otherwise, it's pretty standard after that. Okay. Any questions on that stuff? I'm not sure if he's gonna have you Zoom tomorrow. Like I have no idea what he's gonna do. So if that's something, go ahead, Ani. Did you have a question? Perhaps not. Okay, go ahead.
So would he let us know then before today?
I don't know. Was he planning to have you on Zoom that during tomorrow?
I didn't hear anything, or maybe I didn't pay attention. I don't know if anyone heard that.
Anybody else know?
He usually never does it for like quizzes.
He doesn't do it for quizzes.
No, he just said just go take it.
Okay, well then, maybe just log in at 9:10, just check in with him, and then log out or something. So, yeah. I'll see. Maybe sometimes he'll call me on Wednesday nights, and so we'll see. I'll let y'all know here. Otherwise, so okay. Alrighty then. So we'll just go through amino acids today. So I'm going to really only be focusing on the chemistry aspects of the amino acids. The biology stuff, like sequencing them and all that, you can worry about in biology class. So thankfully there is some overlap here, so I'm really just going to focus on chemical aspects of amino acids. So we'll go from there. Okay, and then so then your exam is tomorrow. Monday we'll have some review. So bring questions. I'm not going to have anything prepared on Monday, so just, bring questions. Take the practice exam so you can get used to the format and the timing of it. And then on Wednesday will be your exam. Final exam, by the way, I'll write this out, is 70 multiple choice questions, and it's 110 minutes. So the practice exam is 7 questions and 11 minutes, so it should be the same timing. But just be aware of that. You'll be able to see all the questions and all that stuff, so you're not going to be locked out of the questions, but of course they will be randomized. And there will be, I actually have a pool of more than 70, so you may not have exactly the same questions, but they should be of similar categories. Like there are categories out like enolate questions, you'll have a certain number of those, and a certain number of ether questions, and blah blah blah. So there's that. The minimal amount of that will be 105 stuff. Most of it will still be 106 stuff. So there's thankfully I have a little bit of control over that. All right. Any questions on the final? That is, I think, the 8th then. And
I don't think you can see your screen.
Oh my god, how embarrassing. Okay. My bad. Well, thankfully all I've done is written all this fun stuff out, so. Okay, so that's on the 8th. Cool, so sorry about that. So now you should be able to see, and you can see all the stuff that I had kind of indicated here. I still didn't check the time. I think it's about 9 o'clock. Is that right? Does that sound right to you guys? We'll know for sure on Monday. I'll double check that. Alrighty. Please note also I posted all of our amino acids there because I'm going to be referencing a lot of these as we go through. So just have that nearby, I suppose, so you can take a look as we go through. Alrighty. Any logistical questions before we do amino acid chemistry?
For exam 4, will it be the same size as the previous ones?
Yes.
Okay.
So about the same amount of stuff as the last ones. Yeah.
Okay, thank you.
Mm-hmm. Yeah, so the only thing that's different is you all get an extra hour for the same amount of material. Alrighty, cool. Anything else? Alrighty, so let's talk about amino acid chemistry here. So first thing we should note is that amino acids have two functional groups, which we hopefully can identify from the name, right? They're going to have amines and carboxylic acids. And we're going to see that if we take a look at the Fisher projections of these things, we're going to have, they're all going to kind of fall under this category, where we are going to have a side chain, which is our R group. We're going to have an amine, and we're going to have a carboxylic acid here. And we're going to notice that all of these have the amine group on the left in the Fisher projection with the carboxylic acid on top. And so these are called L amino acids. So because they have that amine on the left. With all of these, you just have a side chain that varies. You can have it be hydrogen, methyl, all sorts of crazy stuff. There are 20 amino acids that we typically encounter, though there are a few variants on those. And they only differ by the side chain. And so if we take a look at that side chain, if we go ahead and pop over to your, in Discord I've put up the amino acids so we can see them and how they're categorized. We see, of course, in this particular image, we have our amines and carboxylic acids as kind of in the plane there, and then we have all of our side chains on a wedge. So you can take a look at those. We have a couple of categories. I'm just going to start at the top left and we're going to work our way down. So there are 3 amino acids, arginine, histidine, and lysine, that have basic residues. And those bases are amines. You can see they all have extra amines up there on their side chains, which is the top parts of the molecule. And those amines can act as bases. And we're going to see at certain pHs, those amines will be positively charged overall. Because of those extra basic groups, they can grab protons, and once they're protonated, they're positively charged. And if you see those numbers that are next to all those side chains, that's the pKa of that functional group in the corner. We're going to talk about pKa's of the other two, of the amine and carboxylic acid, but we're going to see these first two groups have an additional amine or carboxylic acid that has a pKa of its own. So arginine's pKa for its side chain is 12.1, histidine's 6, and lysine's is 10.7. All right, and then if we see, we also have negatively charged amino acids. Those have carboxylic acids, which will lose their protons, and they're going to become negatively charged overall. So at physiological pH, around 7.4, those carboxylic acids will have predominantly lost their protons. So overall, they will have a negative charge on those side chains. All right, if we go down, so notice, by the way, like aspartic acid, glutamic acid, they only differ by 1 carbon. And so they're fairly similar. The arginine, histidine, and lysine are all fairly different. By the way, of course, you don't have to memorize these. You may have to for biology, but not here at least. So, hooray. So the polar uncharged one, our next category, these have alcohols or amides. So these are capable of doing hydrogen bonding, but they're not capable of losing or gaining protons. And so all of these, like serine and threonine, are alcohol amino acids. They do not have any charge at physiological pH, and they have alcohols, so they can hydrogen bond with things. Asparagine and glutamine look very much like aspartic acid and glutamic acid, except we are switching out the carboxylic acid for an amide, so no ability to be a base here, but they are certainly capable of hydrogen bonding. And they're polar, of course, as well. All right, let's do the special cases, sure. So our special ones, these ones don't really fit in another category. Glycine does not have a side chain, or you can say its side chain is hydrogen. Glycine is the only one that is achiral. All of these other ones are L amino acids, but glycine does not have a chiral center on the amine, so we're not gonna, that's the only one that is not chiral. And proline is special because it has its amine as part of a ring that is connected to the carboxylic acid. So it has some extra bulk there on its side chain. Cysteine has a thiol, so it's got a sulfur on it. It's able to do some special things, as we'll see later on. That sulfur technically is capable of losing its hydrogen. We know that sulfur is a better acid than oxygen is, right? Sulfur is better at accommodating a negative charge than oxygen is. So we do have some minor acidity on that cysteine. And then selenocysteine is a special one because it's the same as cysteine, but it's replaced the sulfur with a selenium. And selenium is even better at holding a negative charge, so actually this one is gonna be technically negatively charged at physiological pH. Although selenocysteine is very, very minor. It is not a common amino acid. It's something in bacteria. It's something that we need in very, very trace amounts. So it's not as popular in our bodies as the rest of them. All right, moving on to the hydrophobic ones. These ones just have alkyl groups as their side chains. So alanine, valine, isoleucine, leucine are all just alkyl groups, and then methionine has a thioether, but that's nonpolar as well. So those have just strictly hydrophobic chains. And then we have aromatic ones as well. So we have phenylalanine, tyrosine, and tryptophan. Those have aromatic side chains. Those are also hydrophobic, but they are special that they are also aromatic. We're going to see that when we talk about cleaving proteins, when we look at these positively charged ones and these hydrophobic aromatic ones, there are certain enzymes that will cleave based on those side chains. So those are special ones for us. Okay, cool. So any questions about the categorization of the amino acids? Alrighty. So let's talk about them at physiological pH then. If we take a given amino acid, let's take glycine because it's the easiest one. We know that this carboxylic acid has got a, I wonder if I have the actual values here. I do. For glycine, the carboxylic acid pKa is about 2.34. Numbers you do not need to memorize. And the amine part has a pKa of 9.6. These are pKa's. Of course, for the amine, that's the protonated version of the amine. So of course, it's not going to just lose its proton normally. Both of these have lower than expected values. Normal carboxylic acids are about 5. Normal amines are about 15. So both of these are low because the two functional groups do affect each other. So they're able to kind of resonate with each other there. So we do end up having both of them be a little bit lower than expected. These pKa values mean that below the pKa, we have the deprotonated form. No, I'm sorry, the protonated form. Below the pKa means more acid. The protonated, protonated form predominates. So if this thing is at pH 1, we would expect the amine to be protonated and the carboxylic acid to be protonated. And so overall, this thing will have a positive charge, right? We sometimes will just draw the amino acid as if there's no charge on it, but we really have to consider what our pH is. If we are above the pKa, we have the deprotonated form predominates. So if we are at pH 12, everything will be deprotonated. So we'll have an overall negative charge on this. And so if you can imagine, if we are at physiological pH, which is 7.4, our carboxylic acid has a pKa of 2.3, so we are going to be above that, meaning we're deprotonating our carboxylic acid, and we're below the pKa of our amine, which means our amine should be protonated. As a result, at pH 7.4, we have the zwitterionic. That's another one that Chalmers is going to test you on your pronunciation skills. So zwitterion. It means you have both positive and negative in the same molecule. So overall, there's no charge because the positive and negative cancel out, but they both are existing. And so we call that a zwitterion. All right, so we at pH 1, we have it protonated. Everything is protonated, so we're going to have an overall positive charge. At pH 12, everything is deprotonated, so we have an overall negative charge. And then at physiological pH, we're going to have a zwitterion, an overall neutral molecule. Something that we can also worry about is called the isoelectric point of an amino acid. And for most amino acids, this is called the Pi, by the way. It will be the average. It'll be your average pKa. I don't know why I'm going to write this as a fraction. So of your groups. So for glycine, the isoelectric point for glycine is going to be 2.34 plus 9.6. And if we, oh my god, I don't have a calculator. Okay, bust out our phone. Actually, I don't have the numbers. Let's calculate this. Very advanced math here. 2.34 plus 9.6 divided by 2, 5.97. So what's special about the isoelectric point is that's the pH at which we have essentially the what's the best way to describe this? It's the pH at which it's completely going to be a neutral molecule. The largest proportion of your neutral form exists. So what this means is, technically at pH 7.4, the predominant form is the neutral zwitterion. I'm sorry, I'm going to move this down. But we know pH 4 is a little bit higher than this isoelectric point. And so we're a little bit closer to that amine one. So maybe we have a few of those amines that are not protonated. A few of them, not very many, maybe 1 out of 1,000 or something is not protonated. But we're going to see that at the isoelectric point, that's where we're going to have the most proportion of our completely neutral molecule. So that's the pH at which you have the largest proportion of your neutral molecule. At these pHs that we've indicated, this is the predominant form, but as we know, with equilibrium and all that, there is some proportion of molecules that are not these at any given time, right? So your isoelectric point is the pH at which most of your molecules, it's your highest proportion of having it completely neutral. If you go above your isoelectric point, you start getting more and more deprotonated ones. If you go below your isoelectric point, you get more and more protonated ones. So your isoelectric point is very useful analytically because each amino acid will have a different one. And it will allow us to separate amino acids. So the point here is it's a measure of our statistics. We're seeing we have the largest proportion of our neutral molecule at your isoelectric point, the Pi, which is just your average pKa.
So quick question. So when you say largest proportion, so there's multiple in there and just most of them are this?
Correct. Yeah. So you have some that have the amine with just 2 protons, some will have the carboxylic acid with a proton, but now most of them have it this way. If we consider what the pKa means, if we consider the carboxylic acid of pKa, that's telling us that at that pKa, at pH 2.34, we have exactly half of our molecules protonated and exactly half are deprotonated. At pH 2.34, you have exactly half. That's what defines the pKa, right? It's your half equivalence point. It's when you have equal amounts of protonated and deprotonated form. So that's why when you go below it, that's when this molecule gets more and more prevalent. And so even at like pH 2, maybe it's going to be 40-60 or something. You're still going to have a balance of the two, but one of them will become more and more predominant. So it just so happens that the Pi, that's when this one happens to be the most predominant. So within that range, between the pKas, you have the largest proportion of your... most of your molecules look like this, within that range. But if you want to skew the balance more and more and more towards having most of them be this form, that's your Pi. Does that kind of make sense more?
Yes, thank you.
Okay. Cool. One more thing. So if the side chain has a pKa, then the Pi is determined by averaging the similar groups. So we'll have a look at what that means with something like lysine. I guess I need to draw this upside down, don't I? So that we're consistent. Okay. I'm going to draw this as H2N. 1, 2, 3, 4, and H2. So lysine actually, does it have 4 carbons or 3? Also doesn't matter. 1, 2, 3, 4, and then they mean 1, 2, 3. Okay. Okay, so here's lysine for us. We have the pKa's of lysine. I have them here, 2.18, 8.95, and then the side chain is 10.53. I know the other one is 10.6. Whatever, close enough. Just use the values that you're given. We're going to see which two groups are similar on lysine. Is that side chain more similar to the amine or more similar to the carboxylic acid? And please don't get this wrong. Which is more similar?
What's similar to the amine?
The amine, right. Yeah, because the side chain is in amine, right? So, of course, that's going to be similar. Or you could just look at the numbers. Which two numbers are the most similar? That's going to be the ones that have the similar groups. So your PI for lysine is going to be those two put together. We don't need to worry about the carboxylic acid. And so this one, let's do the calculation. 8.95 plus 10.53 divided by, or I guess I have to add that first now, divided by 2, is 9.74. So your PI here is the pH at which, again, your zwitterionic form predominates. So we're going to see that with this one, notice that 8.95 is below our isoelectric point. 8.95, remember, if we're going to go above that, that side chain is not going to be protonated. So at pH 9.74, that's when we have that's when this is the predominant form. So our side chain is still protonated, but our amine group is deprotonated. So overall, we have a neutral molecule. We saw our side chain is less acidic because it has the higher pKa. So our side chain is still going to be protonated. Our carboxylic acid and regular amine are both deprotonated, but overall we have a neutral molecule. All right, if we look at physiological pH, we want to look at all of these individually. So this one is going to be lower, so it would be deprotonated. These two are higher, so they're going to be protonated. So at pH 7.4, we have both of them protonated. So overall, at physiological pH, this thing has a positive charge. And its isoelectric point is going to be higher. So the predominant form at all of these pHs is super important because that's what's happening in your body. Right? And so, analytically speaking, it's going to be easier for us to separate lysine from other amino acids because it's got such a high isoelectric point. And we'll talk about that in a second. So, the main things here. Remember, at the isoelectric point, the neutral form predominates. That's its largest proportion. At physiological pH, we need to consider all the pKa's individually. And remember, if your pKa is lower than your pH, you are deprotonated. If you are higher than your pH, if your pKa is higher, you're going to be protonated. And so, if we looked at those numbers again, 8.95 and 10.53, those are both above 7.4. So, at 7.4, both of those should be protonated. and the other one 2 point whatever is going to be less than, so it should be deprotonated. All right, so that's why at physiological pH, this has an overall +1 charge on it. And so that's why we consider it a positively charged amino acid, is because it has a +1 charge at physiological pH. All right. Questions on that? We'll do one for a carboxylic acid. We can do a glutamic acid. Okay, so glutamic acid has how many freaking carbons? 1, 2, and third is carboxylic acid. Okay. One, two, three. Okay, here's glutamic acid. These amino acids, by the way, they all have 3-letter and 1-letter abbreviations. Sometimes the 1-letter, sometimes the 1-letter actually corresponds with the amino acid. Sometimes it's some random letter. Glutamine is not, or glutamic acid is actually an E. It's the amino acid E. So when you're talking about long chains of these things, you might just say 1 letter. Thankfully we don't have to worry about that. That is something you may have to memorize in biology, so just be aware of that. So, pKa's of this thing, of the glutamic acid, this one's 4.25, we have 9.67, and 2.19. So the pI for this, we're comparing now the two carboxylic acids. And so its pI is going to be 4.25 plus 2.19, 3.22. So, if we're looking at physiological pH, we're going to expect both carboxylic acids will be deprotonated and our amine will be protonated. That's based on the individual pKa values of all these. The carboxylic acids are both below, so they will be deprotonated. The amine is above, so it will be protonated. So, we're expecting 1, 2, 3. We're expecting an overall negative charge here, based on the pKa's. So that's why this one is classified as a negatively charged amino acid, because at pH 7.4 it will have a negative overall charge. And if we want to look at the isoelectric point, at that point, notice that our top carboxylic acid is above, which means it will still be protonated. 1, 2, 3. However, our one below will be deprotonated, and our amine here will be protonated. So overall, this is our neutral molecule. And so we see here that we saw with lysine, interestingly enough, it's the side chain that was protonated at the pI. I mean, technically that's the same here, right? Our side chain is protonated at the pI. So it's simply a matter of comparing the pKa's. So 4.25 is the one that's above our pI, so that one should be protonated, and that's going to be the overall neutral one. Okay, so that's the idea here. All right, what's the point of all this stuff? This means we can separate them. So if we're interested in figuring out if you've got a mixture of amino acids to see what amino acids are there, say we're sequencing a peptide, we can separate them based on these PIs. What we do is we'll have a pH gradient gel here. So maybe the pH is increasing as we go from left to right. We're going to have an anode and cathode. And we're going to put our amino acids in the middle. And so maybe this is the high pH side, and this is the low pH side. We're going to see that these amino acids will move based on their PIs. The amino acids will migrate towards either the anode or cathode, until they reach where the isoelectric point equals the pH. So we just dump them all in the middle. We're going to see that negative ones will migrate more that way. Positive ones will migrate more that way, until we end up reaching their isoelectric points. So they're going to keep moving along this pH gradient because they'll be charged. Actually, I've got these two. This should be low pH here. This one should be high pH. So as this, say we have this positive amino acid moving over towards the cathode. It's moving towards the negative side because it's attracted to that. And eventually it reaches a pH at which it equals the PI. So maybe right here is our pH equals to, what was our PI for this one? 9.74. At that point, our amino acid goes, our lysine goes there, and at that point it stops. Why would it stop there? Well, remember that 9.74 is the pH at which it's now neutral. If it's neutral, it is no longer attracted to the cathode. Remember, positive things would be attracted to the negative things, but as soon as it's no longer positive, it's not going to move anymore. So they're going to keep moving until they reach their PI. And so we can separate amino acids this way. If we keep going to this side, what's our glutamate 3.22? Our glutamate will migrate until it reaches the point at which pH is 3.22, and it's going to stop there. So if we, for example, had lysine and glutamate together, they will both migrate to opposite sides and we will have separated them. If we had our dear friend glycine in here, that was 5.9, I think. I forget. 5.97, okay. So maybe our glycine will move now over to here. So we have our glycine there. So we have separated all three of them. All right, so they will just move to wherever their pH happens to equal the PI, and they'll move either towards the anode or cathode, depending if they're positively charged or negatively charged once you put them in the middle there. All right, so we can separate them. This is called electrophoresis. Alrighty. So, any questions on separating them?
So electrophoresis is the separation.
That's
of them.
this method is called electrophoresis, yeah. So it is this analytical technique. All right. By the way, how do you tell where your amino acids are? So this is not really suitable for many amino acids. It's for smaller mixtures. You can detect them by using Ninhydrine. So Ninhydrine is this molecule here. Too many carbons. This is Ninhydrine. And it will form a purple product if you combine it with an amino acid in basic conditions. So whatever your amino acid happens to be, you'll end up getting, it doesn't really matter what your amino acid is, but you end up getting the following here. You end up putting two of these ninhydrines together. Oh no, I might make out of space. Oh no. Pretend that's a phenyl group on that side. This thing is purple. Amino acids, ninhydrin themselves are colorless, but you, if you have the amine there, an amine can be detected with ninhydrin. It'll produce this purple thing. And so you can see, therefore, on your electrophoresis where your amino acids are by dyeing them purple with this. You'll end up with a bunch of side products, but who cares? The point of this is detection. And so you can detect your amino acids. You can find out where they are. And then from knowing where those pHs are, you can figure out what amino acids you have. Because it doesn't matter which one, right? Whatever is at that PI has to be a certain amino acid. All amino acids have different PIs, so you can separate them. And once you know which pH you have, at your given dot, whatever this pH is, you can figure out if there's lysine in your mixture, it will show up at a purple dot at 9.74. And so if it's there or not, that's the purpose of this purple stuff. So you can tell if you have lysine or not. This is not a quantitative measurement. You can't say how much lysine you have, but you can say, do I have it or not? And that's using this. So this is how you detect them. All right, so essentially this thing just condenses on both sides, and you end up with a purple product. Because this thing is fully conjugated, and it's got the nitrogen in there. That's how we make colorful things. All right. Cool. Now let's work on the synthesis of amino acids. We have a couple of ways. They're all crap, but we can do them if we are so inclined. If we want to make an amino acid, we can either start with the carboxylic acid or the amine, and we can go ahead and attempt to make them. What we can do, one way to do it, what's this reaction, by the way? What does this do? This is one from the Untitled chapter. Perhaps you remember this. Got a fancy name for it. This is our Helmholtz-Zielinski reaction. It brominates the alpha position of a carboxylic acid. Okay, good. I'm glad you've completely forgotten. Excellent. Okay. And then if you just throw excess amine at this thing, you can get it to replace the... Oops. You can get it to replace the bromine. However, of course, this will be racemic because this is also racemic. And the reason you're throwing excess amine at this is you're going to try and prevent polyalkylation of your amine by just throwing a ton of it. And so not only will sterics help here, but just the excess of it will hopefully prevent you from polyalkylating your amines. Alright, so you can make an amino acid that way, but it's racemic, so it's useless. Alright. We can also do something interesting. We can do another kind of modified clays in here. If we take a protected amine and we can just go ahead and use our alpha proton chemistry here. Remember that alpha proton is going to be pretty acidic. If we treat this with a base and an alkyl halide, we're going to end up alkylating at the alpha position. You'll end up with your R group there. And then you can just decarboxylate this thing and you'll end up with an amino acid. It's going to decarboxylate one of those sides. You're going to end up with your amino acid. Oops. And of course, guess what? This is racemic, so also useless. It's actually quite a lot easier to just harvest amino acids from biological organisms, but if you were so inclined to make them synthetically, you could. Although, as you see here, all of these are racemic, so it's not so great. Alright. So we can do that. Decarboxylate a type of, what is this, diethylmalonate kind of synthesis. Similar to there. We can do the Strecker. You take an aldehyde. You're going to treat it with ammonium chloride and cyanide. You'll end up replacing both, or you'll end up getting both of those on here. So you end up with an amine and a cyano, a nitrile on there. And as we know, we can just hydrolyze the nitrile, and we'll end up with our carboxylic acid. And we have our R group. Guess what? Racemic. So, just like before, we'll end up with a racemic mixture here. So, not so great. There are ways to get certain amino acids enantiose selectively, but you've got to use chiral catalysts and all that sort of thing, so we're not going to really focus on it too much because these are beyond the scope of the course. So the point is, it is kind of quite silly to try and synthesize amino acids when you have biological organisms that have these things in great amounts, and you can just grow some cells up and take and just isolate the amino acids and purify them. So, doesn't really make sense. But you can. Cool. All right, let's take a look at various ways to cleave peptides. Peptides are multiple amino acids together. And so we can cleave them. Two ways to do it. We can chemically cleave them, which is a pain, or we can use enzymes to do it. So in terms of chemical cleavage, we have the Edman degradation. One day I'll learn how to spell degradation. Here we go. So you've got your other amino acids. And we're going to have our R group here. So this is a peptide bond. This is from a carboxy group of one amino acid and the amine of another amino acid. Those have come together to make an amide. So remember that carboxylic acid plus an amine can give you an amide. So that's the bond that we see there. That's the peptide. So it's the carboxylic acid from one amino acid, the amine from a second amino acid. Putting those together, we get an amide between the two. And so we can cleave that by using phenyl isothiocyanate. This is an isothiocyanate. And then we use trifluoroacetic acid. And what will end up happening is we're going to get our R group separated from our new compound with all of these from the phenyl isothiocyanate. So this thing you can detect if you are so inclined. You can detect it. You don't really have to worry about what this means. It means we can cleave off a peptide. That's the point of doing this. So first you treat it with the isothiocyanate. Once you treat it with that, treat it with trifluoroacetic acid, you will cleave off one peptide. And so that one peptide can be analyzed. And you can find out what that peptide happens to be. And then you'll have the rest of the peptide will be ready to go, and then you can repeat this process. And so you can keep doing it over and over and over again, and isolating and analyzing each of these crazy heterocycles that you get out. So you can analyze that, figure out what amino acid it came from, and that'll tell you what the order of amino acids is. So by cleaving them one at a time and analyzing the products, you'll figure out what your sequence is. It's rather tedious, isn't it? Imagine if you have a chain of 50 amino acids, you're going to have to do 100 reactions to sequence it, or 98 technically, but the point is tedious, right? There are better ways. We can use enzymes. We have trypsin and chymotrypsin. These cleave amino acids. They're enzymes, so they're very specific. They will cleave the carboxy terminus. They'll cleave the carboxy side of lysine and arginine. so our basic amino acids. And chymotrypsin cleaves the carboxy of tryptophan, tyrosine, and phenylalanine, are aromatic ones. So it cleaves them. And so we can use this information to figure out what an amino acid might have for us. So if we take a look at, these are the two enzymes we use, by the way. If we take a look at a sequence, let's write all these out. Proline, methionine, tyrosine, lysine, arginine, serine, tryptophan, leucine, and histidine. So here's a small peptide. If you wanted to do admin on this, you could. It would take however many there are. There are -1 times 2 reactions. Pain in the butt. Or you can use the enzymes. And we're going to see that the trypsin is going to cleave after lysine and arginine. And the chymotrypsin, this will be the blue one, this will be the purple one, will cleave these afterwards. So we're going to end up with 2 sets of fragments. We'll take a look at those. Let's see, we've got a phenylalanine, tryptophan, and a tyrosine in here. So if we treat this with trypsin, we end up with the following fragments. We get alanine, phenylalanine, and lysine. That's one fragment. We have proline, methionine, tyrosine. We have more glycine and then arginine. And then we have serine, tryptophan, leucine, histidine. We have, these are the fragments that we'll get by treating it with trypsin. So we cleave after the basic residues. Not including histidine. We don't cleave after histidine. So just lysine and arginine. With chymotrypsin, these are the fragments we get. We get alanine, phenylalanine. We get lysine, proline, methionine, tyrosine. We get glycine, arginine, serine, tryptophan, and then leucine, histidine. So, with this, we're going to need to combine the information we get from both enzyme treatments. So notice we have alanine, phenylalanine, lysine, but on chymotrypsin we just have alanine, phenylalanine. So we know from the trypsin that lysine must follow phenylalanine. And since we only have one other lysine, that means this part must follow the phenylalanine. And so it's simply a matter of using information from both of these to put this together. So because we know this lysine comes after phenylalanine, because we see that in the trypsin treatment. We know that these fragments must be connected together. We have only one other lysine here. And so that tells us that we at least have that part of the sequence. And then if we take a look at the rest of them, we see, this is an easy example because they're already in order for us. But if we see this tyrosine, we see that, well, the tyrosine must be between methionine and glycine. So we now know that the glycine is going to come after the tyrosine. And then we have tryptophan here, and so we know that the leucine comes after the tryptophan. And so it's a little bit easy right now because I've given them in order. Let's do another one with they're not in order, and we can see what we're going to end up with here. We'll see if we can't assemble the protein here. So we're going to use information from both enzyme digests to figure it out. So here's a problem. From cleaving with trypsin, remember that's lysine and arginine that it cleaves after. Here are the fragments we get from trypsin. Alanine, glycine, phenylalanine, lysine. Isoleucine, tryptophan, methionine, histidine, phenylalanine, methionine, cysteine, arginine. These are too big. Oh, well, we'll be able to fit it. And then if we look at the chymotrypsin fragments, methionine, histidine, phenylalanine, methionine, cysteine, arginine. I'm gonna write this one down below just because it's got more stuff on it. Arginine, alanine, serine, alanine, glycine, phenylalanine. Okay, sorry about that. Okay, there we go. Gosh, that took a long time to write. Chymotrypsin cleaves off phenylalanine, tyrosine, and tryptophan. Let's see what we can do to put these together. So, we know that trypsin cleaves after lysine and arginine. Chymotrypsin cleaves after phenylalanine, tyrosine, and tryptophan. So, hopefully, we can tell which amino acid fragments belong to the last ones. So remember that everything after a lysine or arginine will be cleaved off with trypsin. Everything after an aromatic one will be cleaved off with chymotrypsin. So we should be able to first identify the end of our amino acid, the carboxy terminus, the last ones. What here does not have something that cleaves after it? Let me rephrase that. There's a fragment here that has to be the end of our amino acid. We know that because there are no cleavage. There is no cleavage after that particular amino acid that we can do with these enzymes.
Is it the one with the G?
Is it the one with the G?
The G?
G.
I don't know which one you mean.
Oh, the glyc.
Oh, the
glycine. No. So that amino acid is not at the end, right? Because this one's in the middle. It's already in between two amino acids. So which amino acid has to be the last one? So really, our options here are these ones. So which of these amino acids must be our last one? It's the cysteine, right? And you know that because there's nothing that comes after it. We know that trypsin cleaves after lysine arginine. Chymotrypsin cleaves after the aromatic ones. Nothing cleaves after cysteine. So if we have a fragment that ends in cysteine, that has to be the end of the protein. because there's nothing that's going to cleave after cysteine. So if we have a cysteine at the end, there's nothing else that could have followed it. So we know the end of our amino acid must be we know that it must be this lysine, proline, cysteine. How do we know that? Proline, cysteine, lysine, proline, cysteine. We know that the trypsin will have cleaved that lysine off, but we know that that must be the last three, right? Because we see that fragment in the chymotrypsin. So we know that lysine comes before proline, and we know cysteine is the last one. So what comes before the lysine then? What's the amino acid that has to be in this spot? Looking at these fragments.
Oh,
It kind of
by
goes
order, so it's
by
like-
order, so it's like
That's
so if
so it has
that's
to be it.
so it has to be it.
So it has to be the one.
So it
Just
has
say
to
the
be
letters.
the one.
It's fine.
The chymo.
Or like, go ahead. Yeah.
So it would be the.
So it would be the
TRP one?
TRP one.
Which one? This one?
So it would be one from the.
So it would be
right side?
one from the right side.
Could be either. So, because these both, it's the same peptide being cleaved in different ways by different enzymes. So, we have from this one, this has told us that this is the end fragment. So we don't need this anymore. We don't need this anymore. We've already taken care of that part of the protein. So we're trying to figure out what's coming before the lysine in this. And so here's a hint. There's only one fragment that even has a lysine still here.
It's just the top one.
The top
it's
one,
just the top
yeah.
one.
So that means that this lysine must be here. So that means before this comes the phenylalanine, the glycine, the alanine, the serine, the other alanine. Do we see how we got that?
Yeah. Yeah.
So, with these, we know that this lysine, there's only one lysine in this protein. We already know what comes before that lysine because we have a fragment that shows us this. And so now we have to think, okay, well, we're at this alanine. What comes before the alanine? So this fragment is, we've already taken care of it. We don't need that anymore. So what comes before this alanine? So we should look for fragments that have alanine. Hopefully we can see there's only one.
So
Go
it
ahead.
would be the last.
The last
It
one,
would
right?
be the last
Yeah,
one
right.
on the
So
right.
we have all this stuff. The alanine, serine, alanine, glycine, phenylalanine. We have that right here. So we must have arginine, cysteine, methionine coming before that. I don't know how I'm going to do this. Let's see. Let's move. Oh, God. So we know before the alanine... Oh god, I don't want to write this backwards. Well, I'm going to do it anyway. We know that arginine... this is backwards order on the top, just because I'm out of space. So, but we know that this is the connection. Arginine comes before alanine, cysteine comes before arginine, methionine comes before cysteine. So, this fragment's done with. What's the next fragment that we're going to be using?
So bottom left.
Bottom
So
left,
bottom
right, because we see we have these three here. That's these three. So that means that next we have phenylalanine, histidine, methionine, tryptophan. Oops. God, this is such a mess. Tryptophan, isoleucine. And in fact, that's the entire thing. That's it. We're done. So this is our protein that we're able to sequence together by looking at the cleavages that we saw. Because the isoleucine tryptophan, that's already taken care of. It's right here. The methionine, histidine, phenylalanine is right here. We don't need to worry about those fragments. Or do another one. Yeah, go ahead.
So for the
We're
connecting it.
so when we're connecting it to the
So
ALA one, so
where does it?
So this will be the
You did it
first
from
one,
arginine.
and this is the last one. I'm totally like, I realize there's all these scratches everywhere. This is our first amino acid here, is the isoleucine. And our last amino acid is the cysteine. So that's the order. It goes isoleucine, tryptophan, methionine, histidine, phenylalanine, etc., all the way to cysteine. So just because of the fact that I ran out of space, I had to do that top one kind of backwards. So, I mean, I apologize for that. Not easy to do these, right? So maybe the next one we can do vertically or something. We'll have more space.
Then for whenever it starts from
ALA, why doesn't it go from SERALARG CYR?
Oh,
It's backwards.
Oh.
That's why.
okay.
Yeah. It's just
okay,
because
okay.
it's yeah. Sorry about that. It's just the fact that it's backwards. So sorry. All right, let's do one more of these, and then we'll be good. So here's the trypsin ones. Oh my gosh. This is a longer one, but that's okay. Okay, so those are the trypsins. The chymotrypsins. Tyrosine, methionine, cysteine, arginine. Okay. Took 10 minutes to write out the problem, but we can go ahead and sequence this. So as before, I want to start by finding the last one. So what's our last amino acid going to be?
Would it be? I don't know what it's called.
Would it be? I don't know what it's
Yeah,
called.
valine.
The
You're
VAL
absolutely
one.
right. And how did you know that? Because it's the only one that's not arginine, lysine, or an aromatic. Right? It's the only one that is, it's going to be, this is our end fragment here, the valine. So it's the only one that is not an aromatic or basic amino acid residue. That's the end of a fragment. So that means we have alanine, valine, and in fact we can go ahead, because we know that that fragment is here, we can go ahead and put these 5 as our final fragment. PHE, proline, glycine. So we've taken care of this fragment, taken care of that fragment. Generally speaking, by the way, you will usually bounce between the two. So like after you use a trypsin one, then take a look at the chymotrypsin ones, and then take a look at the trypsin ones again. So we just had gly, pro, and phenylalanine. So which chymotrypsin one has glyprophenylalanine in it?
Bottom left.
Bottom left.
Bottom left.
So that's great. So this means that these correspond with these 3. So what comes before must be arginine, cysteine, methionine. So now we go up here. Which one has methionine, cysteine, arginine in it?
Top left, right?
Top left.
Top left, right?
Yeah, first one, right. So we already have the methionine, cysteine, arginine, so now we need PHE, his, and tryptophan. I'm going to write it this way, so it may be a little bit easier. So I'm going to write it that way, so backwards. So okay, so we have, now this one's taken care of. So now we're looking for tryptophan histidine here. Or more likely histidine phenylalanine. But that's already taken care of. We don't really need that. It's already in our thing. So we need something that has a tryptophan in it.
The
First one,
first
yes.
one in-
So we have arginine, leucine, isoleucine, valine, proline, lysine. So that's taken care of. So we already see this proval, isoleucine, leucine, arginine already in our thing. We already have that whole sequence in here, so we don't need it. So we're looking at something with lysine. And hopefully we can figure that out, because it's the only one left. Okay, so great. So we must have tyrosine before that. Then alanine, valine, phenylalanine, methionine. Alright, and of course val-ala-tyre is already in here. So we don't need to worry about that. Smaller ones are typically, and methionine, phenylalanine, that's the first segment, we don't need that. Usually the smaller ones are less useful, but we will have fully sequenced our thing. So, ta-da! We have sequenced it. So, starts at methionine, ends at valine. This we can call the N-terminus, the first one. And then the C-terminus, which is our last one. So, hooray! Okay, so that's it for today. We're good. Okay, so that's it. tomorrow 9:30 your exam. We technically haven't finished this, but whatever. We're fine. I will make sure that we don't have anything past this on our final. So, okay. So tomorrow, good luck on your exam. Monday we'll review, and Wednesday will be your final. So it's been fun. Bring questions on Monday, okay?